Hello everyone. I welcome you all to lecture 1 on animal kingdom. So basically, till the last lecture, we learn about the biological classification, what is the basis of classification, and how different organisms are classified into different kingdoms. So in last lectures, we learn about kingdom Monera and kingdom Protista, and in today's lecture, we will learn about kingdom Animal. So, according to the R.H. Whitaker classification, all the prokaryotes were classified into the kingdom Monera, while all the eukaryotes were classified into different kingdoms like kingdom Protista, Plantae, Fungi, and Animalia. So, now on the basis of the different modes of nutrition, all the eukaryotes were further divided into the different kingdoms. And today we are going to learn about the characteristics of kingdom Animalia. So animals can be contrasted with plants and fungi. Like as I mentioned that all like Protista, Plantae and Fungi, Animalia group includes the organisms that are eukaryotic in nature. So like both of these, that is like both plants and animals, like both plants and fungi, animals are multicellular eukaryotes. But unlike plants, which are autotrophic in nature, that is, they can make their own food through the process of photosynthesis, uh, animals are heterotroph. That is, they must acquire their nutrients from an external source due to the lack of the pigments like photosynthetic pigments like chlorophyll. These organisms are not able to synthesize their own food, and therefore they belong to the category of heterotrophs. So just like plants, they are multicellular eukaryotes, but they differ from the plants on the basis of their mode of nutrition. In plants, the mode of nutrition is basically they are autotrophic, while the organisms that belongs to kingdom Animalia are heterotrophic in nature. That is, they cannot synthesize their own food and therefore they are dependent on the external source, or they must acquire their nutrients from some other sources. Now, if I compare organisms of kingdom Animalia with the organisms of kingdom Fungi, both of them are mostly heterotrophic in nature. That is, both cannot synthesize their own food, but the mode of digestion differs in both. For example, if I talk about the organisms that belong to kingdom Fungi, they digest their food externally. That is, they release the enzymes outside the body, leading to the degradation of the components, and then they absorb those products. While if we talk about the digestion in animals, so animals ingest, that is, they eat their whole food and then they digest it internally with the help of enzymes. So this is the difference in the how the organisms that belong to kingdom fungi obtain their nutrition from the food, while how the organisms that belong to kingdom Animalia obtain their nutrition from the food. So the organisms that belong to kingdom fungi, they first break down their food into simpler products and then they absorb the food. While if we talk about the organisms that belong to kingdom Animalia, they ingest or eat the whole food and then they digest it internally with the help of enzymes. So in this, the digestion is internal, while if we talk about the organisms that belong to kingdom fungi, the food is digested externally. So these are the differences between the organisms belonging to different groups of Plantae, Animalia, and fungi. While the organisms belonging to all these three kingdoms are multicellular eukaryotes, yet there are differences in the mode of nutrition or in the uptake of nutrition from each other. Next, we can say that animals have diploid life cycle. So what do I mean by diploid life cycle? So in diploid life cycle, the individual is always 2N. And meiosis results in the production of gametes. Gametes are N in nature. And basically the fertilization, that is by the process of fertilization where the fusion of gametes takes place, that is two haploid gametes which are N and N in nature, fuses to form the zygote that develops into an individual which is 2N in nature. So basically the animals have diploid life cycle, that is throughout their life cycle they have 2N number of chromosomes and they usually carry out sexual reproduction and begin life only as a fertilized diploid egg, which is known as the zygote. So zygote is nothing but a fertilized diploid egg which has been developed, and it is 2N in nature. So the fertilization of two haploid gametes result into the formation of a zygote, which is 2N. And this, upon development, leads to the formation of the complete individual, which is again 2N in nature, which is diploid. And during the fertilization, meiosis happens, leading to the formation of gametes, which are haploid, which are N in number, and again the fusion of these gametes leads to the formation of zygote. So the life cycle is diploid, individual is always 2N, and meiosis results in the production of gametes. From this starting point, that is from the fertilized diploid egg or the zygote, they undergo a series of developmental stages to produce an organism that has specialized tissues, usually within organs that carry on specific functions. So as I mentioned, that zygote undergo development, and during these developmental stages, it can produce some specialized tissues, or these tissues may develop into some specialized organs that carry on specific functions. So depending on the level of organization, zygote develops into different stages, like it may develop into specialized tissues. These tissues may further develop into specialized organs, etc. So this was about the life cycle of the animals. So animals follow the diploid life cycle, in which the individual is 2N in nature. So what is the basis of classification of different animals into the different groups? So the complete animal kingdom, the organisms belonging to complete animal kingdom can be classified into different groups on the basis of following. So the first is the level of organization, that is how the body is built, like whether it is made up of cells only, it is made up of tissues, or it has an organ level of organization or organ system level of organization. The second is metamerism. So metamerism means segmentation, that is whether the body is segmented or whether it is not segmented. The third is body cavity, whether the coelom, body cavity, which is also known as coelom, is present or coelom is absent in them. Next, we can divide the organisms on the basis of their symmetry, that how the body can be divided into two equal halves. So whether they follow a radial pattern of symmetry, or do they have a bilateral form of symmetry. Based on that, the organisms can be divided into the different groups. Next, we have on the basis of division on the basis of germinal layers, that is the layers which are formed during the development stages. So there are three germinal layers usually: ectoderm, mesoderm, and endoderm. While in some organisms only two are present, so on the basis of that also organisms are classified. Next, we have notochord, whether the notochord is present or not present, and then we can divide the organisms on the basis of circulatory system also, like whether the open circulatory system is present or whether the circulatory system is of closed type. So we will learn about all of these bases of classification one by one in detail. So starting firstly with the level of organization. So there are different levels of organization, as we can see in this diagram. So a small molecule might be present as a level of organization. These small molecules may interact among themselves, resulting in the formation of larger molecules. These larger molecules may be present as a component of a cell. So this is the figure of an animal cell. We know that animal cells are eukaryotic in nature. That is, they have membrane-bound organelles and they have a well-defined nucleus. So basically, so this is about the eukaryotic cells. Animal cells are eukaryotic in nature, and after cells we have tissue level of organization. So certain cells acquire a similar type of function, results into the tissue formation. For example, here I have represented the cardiac or the heart tissue that is present in the heart cell. Next, tissues results in the formation of the organs that have a specialized function. For example, heart. So it pumps the blood all over the body. These organs may be a part of a complete organ system. For example, cardiovascular system. So heart is a part of a cardiovascular system where along with different organs like blood, like this interact with the blood vessels and supplies the blood to the complete body. So this is a part of a organ system, and such different types of organ system lead to the formation of a complex organism. So like we have a circulatory system, we have a digestive system, we have nervous system. So these different organ systems are present that contains different organs. For example, heart is a part of cardiovascular system, brain is a part of nervous system. So these different organs result into the formation of a particular organ system, and these organ systems interact among themselves, leading to the formation of a complex, complete organism. So this is the level of organization that starts from the small molecule and results in the formation of the complete complex organism. So on the basis of the level of organization, we can divide all the animals into different groups. The first is a protoplasmic level. For example, in protozoans. So we have learned about the protozoans in the last lecture. So they have the protoplasmic level of organization. So the unicellular body performs the all biological activities. They do not have any level of organization at the cell level. The next is the cellular level. So we will learn later also that in sponges, which are a group of animals, cells are arranged as loose cell aggregates. So basically these cells are arranged as loose cell aggregates, and division of labor occurs among the cells only. There is no tissue which is present. So no tissues are formed, and therefore the level of organization is the cellular level, where the division of labor, that is the function, is divided among the different cells only, and no specialized group of cells is present, that is no special tissue-like structure is present. The next is tissue level. So in coelentrates and ctenophores, cells performing the same function are arranged into the tissue. For example, the cells that have the same function of maybe the beating results in the formation of the heart tissue. So similarly, in coelentrates and ctenophores, all the cells that perform the similar kind of function are arranged into the tissues, resulting into the tissue level of organization. So you don't need to worry about these things much now, because when we will learn about these different groups one by one in details, we will again discuss all these points there, that how in sponges there is a cellular level of organization, how a particular cell, how the division of labor is taking place at the cellular level of organization, or how in coelentrates and ctenophores cells are performing the same function, are arranged into the tissues. Now, in platyhelminthes, tissues are arranged to form organs, and therefore we have an organ level of organization there. After that, in higher animals, that is from SK helminthes to chordata. SK helminthes, annelids, arthropods, mollusca, echinodermata, hemichordata, and chordata, the level of organization is organ system level. That is, in higher animals, organs are further organized to form the organ systems. So, in platyhelminthes, there is no organ system that is present. While if we talk about higher animals, that is from SK helminthes to chordata, the organ system level of organization is present. So in higher animals, organs are further organized to form the organ system, and therefore they have organ system level of organization. So when we will learn about these groups one by one in detail, we will discuss these points again there. But for now, on the basis of level of organization, all the animals were divided into different categories like protoplasmic level, cellular level, tissue level, organ level, and organ system level. Next, which is a very important classification system, is that is used as symmetry. So organisms may be asymmetrical, or they may have a radial symmetry, or they may have a bilateral symmetry. Depending on the type, how the cells are arranged, or how the body can be divided into two equal or similar parts, it defines the type of symmetry the organism has. So when the body cannot be divided into two similar parts, the organism is said to be asymmetrical. Like there are certain substances or certain organisms that cannot be divided into two equal parts, no matter on what axis we will use, whatever axis we are using to divide the organism into two similar parts. So such organisms are known as asymmetrical in nature. That is, they do not have two similar parts irrespective of whatever axis we are using. For example, most of the poriferans are basically sponges. So most of the sponges are asymmetrical in nature. The next is a kind of symmetry which is known as radial symmetry. So as the figure also demonstrates, in radial symmetry you can use any plane. So any plane which passes through the central axis of the body. So this may be plane number 1. This may be plane number 2. This may be plane number 3. So any plane that passes through the center of the body divides the organism into two identical halves. For example, some of the poriferans, cnidarians, ctenophores, and adult echinoderms. So this is very important because in echinoderms, the symmetry is of two types depending on whether it is an adult echinoderm or if it is an adult echinoderm, we will answer it has a radial symmetry, but if we are talked about normal echinoderms, we will answer the bilateral symmetry. So in bilateral symmetry, the body is divided into identical right and left half only. We have our one half at the right side and the other half on the left side. And unlike the radial symmetry where any axis that passes through the center of the body divides the organism into two identical halves, in bilateral symmetry we only have one axis that divides the organism into the left and right side. So for example, if I consider this as an axis of symmetry, it does not divide the organism into two equal halves, and therefore it only has one axis of symmetry that divides the organism into two identical right and left halves in only one plane. So examples include platyhelminthes to chordata, except as I mentioned, adult echinoderms. Adult echinoderms have radial symmetry. This is very important exception, and this we need to remember because usually we are asked questions about the symmetry. So this is a very common question that is being asked, that about the adult echinoderms which have radial symmetry, unlike the other organisms from platyhelminthes to chordata that have bilateral symmetry. So this figure explains radial symmetry in much more details. Sea animals that belongs to ctenophores, for example, they have on the top side, they have a top side where mouth is located. So this is the top side and this is the bottom side. So they have a top side where mouth is located, while they all have a bottom and they have a bottom side. In such cases, they do not have any front or back side, or no left or right side. For example, if I consider this as a flower pot, so we do not have any front or back or right or left side to that flower pot. We just have a top and a bottom, and any plane that passes through the center of that flower pot will divide it into the two equal halves. Irrespective of, since we do not have any right or left and front or back system in such organisms, we just have a top and a bottom system. So any plane that passes through the center of such organisms forms a plane of symmetry, and this kind of symmetry is known as the radial symmetry. So in a radial animal, for example, such as sea animal that belongs to the phylum Cnidaria, they does not have a left side or a right side, and therefore any imaginary slice or any imaginary plane to the central axis divides the animal into mirror images. So this is about the radial symmetry. The next is a bilateral symmetry. So in bilateral symmetry, we can consider a bilateral animal such as lobster, which belongs to the phylum Arthropod. It has a left side and a right side. So we can see that if we use this plane as a plane of symmetry, we will not get the two similar equal halves. While it just have one plane of symmetry, so one is the left side and the other is the right side. So the plane of symmetry divides the organism into two equal halves on the basis of their left and right side. So only one imaginary cut can divide the animal into mirror image halves. So, for example, we can use this example where we can see that only one imaginary cut can divide this component into two equal halves. The one is the left side and the other is the right side. Even in humans, if we will try to understand using the example of human, we'll have just one plane of symmetry that passes through the center of the body that divides the organism into the mirror images as left and right side. So therefore, a bilateral animal has one imaginary cut, only one plane of symmetry, and we can say that it has two axes of orientation. So there is a difference. I am talking about one plane of symmetry and two axes of orientation. So unlike the organisms that have bilateral symmetry, that have radial symmetry, where there is no front or back or no left and right side, in case of bilateral symmetry, we have front to back and top to bottom orientation. So in such organisms, we have front to back orientation also and top to bottom orientation also. So the top side is known as the dorsal side, and the bottom side, we have a ventral side. So such animals have a dorsal top side and a ventral bottom side. And also, as I mentioned, they have a right side and a left side. Again, we can say that they have an anterior front end. So the front end is known as the anterior end. So the part which is represented here in yellow color is the anterior end, while the part which is represented here in the red color is the posterior end. So which is the back end. So many animals with bilateral symmetric body plans, such as arthropods and mammals, have sensory equipment concentrated at their anterior end. So most of the organisms, like even in humans, we have brain which is present at the anterior end. So most of the sensory equipments are concentrated at the anterior end, including a central nervous system, that in example brain in the head, which is present on the anterior side, not on the posterior side. So basically, this was about the bilateral symmetry, where only one imaginary cut divides the animal into two mirror images. And bilateral symmetry has two axes of orientation, as I mentioned, dorsal and ventral. We can say or anterior or posterior. So dorsal and ventral is top side and bottom side, while if I talk about the anterior and posterior, it is the front and the back end. So the symmetry of animal is very important, and it generally fits its lifestyle. So why a particular organism has adopted to that particular symmetry is because of its lifestyle. For example, many radial animals are sessile. That is, they do not move. They live, usually they spend their life attaching to a substrate. So they usually live while they are attached to a substrate. Or they may be planktonic. That is, they drift or weakly swim, for example, the jellyfish. So they may be sessile or they may be planktonic. So since they have a radial symmetry, this symmetry actually helps them to meet the environment equally well from all the sides. Like for example, in radial animals, like for example in sea animals, we can see that these organisms are usually sessile or they are planktonic, that is, they drift in the water. So such organisms require the force that, or they require the, they should have a body plan in such a way that they can sense the environment equally well from all the sides. So therefore, they have a body type which have a radial symmetry. While if we talk about the bilateral animals, they typically move actively from place to place, and therefore they may face differences in the environmental conditions from the front side or the back side. So most bilateral animals, as I mentioned, they have a central nervous system that enables them to coordinate the complex movement involved in crawling, burrowing, flying, etc. And therefore, they do not require the body shape or the structure that is radial, that should have a radial symmetry. So this is how the symmetry plays an important role and how it fits into the lifestyle of a particular organism. So the radial symmetry actually allows the organism to sense the environment or to interact with the environment equally from all the sides, as it cannot move much and therefore it has to remain in that environment almost all the time. Therefore, it should be able to sense the environment from all the sides equally. While if I talk about the bilateral animals that are actively moving from place to place, they have different organs which are concentrated at different sites. For example, in most of the bilateral animals, the nervous system or the CNS, like the head, which is the controlling unit, is usually located at the anterior end of the organism. The next basis of classification is germinal layers. So germinal layers are the ones which are formed when the organism is undergoing the embryonic development. So for example, like all animals, sponges are multicellular, but they do not have any true tissue. So when we were studying about the level of organization, we studied that sponges have a cellular level of organization. That is, they do not have tissues. The division of labor occurs among the cells only. So therefore, we can say that the sponges have a cellular level of organization. Now, the organisms which have true tissues, that is from the coelentrates to chordates, the organisms which have true tissues, they undergo the embryological development. That is, their embryo undergoes the development, and during the development process, different tissue layers are being formed. So the first three tissue layers which are formed are often called the germ layers. Why they are called the germ layers? Because during the process of development, these layers give rise to the organs and organ system of the complex animals. So these organisms are the first three layers. These layers are the first three layers which are formed. So these three tissue layers are called as the germ layers, and later on these three germ layers are the ones that are responsible for giving rise to the formation of organs or organ system in the complex animals. So for example, animals such as cnidarians, that is coelenterate, which have only two tissue layers like ectoderm and endoderm as embryos, are known as diploblastic with the tissue level of organization. So basically, the organisms like belonging to the cnidarians. So the cnidarians are also known as diploblastic. That is, they have only two germ layers. On the bits of germ layers, as I mentioned, the organisms were divided into diploblastic and triploblastic. That is, the ones which have two germ layers and the ones which have three germ layers. So those animals which have two germ layers are known as the diploblastic. They have tissue level of organization. They have only ectoderm and endoderm with no mesoderm present in them. While those animals that develop further have all the three tissue layers, that is the ectoderm, mesoderm and endoderm. So as the name suggests, ecto means outer, meso means the middle one, and the endo means the inner germ layer. Are triploblastic in nature, and as I mentioned that these three germ layers together give rise to different organs and organ system of complex animals. Therefore, it is present in the organ level of organization. So we can relate these things from each other. Like as I mentioned, we can relate symmetry to their lifestyle. Similarly, we can relate the level of organization with the germinal layers that are being formed at the embryonic level. So in the cellular level of organization, we do not have any germ layers, while the ones which have tissue level of organization, they may have either two germ layers, that is ectoderm and endoderm, and therefore they are diploblastic, while if they have three germ layers, that is all the three germ layers which are tissue layers which are being formed, that is ectoderm, mesoderm and endoderm. In such cases, the embryo is known as triploblastic, and they have organ level of organization where these three tissue layers lead to the formation of organs. So ectoderm, what is ectoderm, mesoderm and endoderm? So ectoderm, as I mentioned, is the outer covering of the animal, and in some phyla it leads to the formation of the central nervous system. So it is the outer covering of the animal. It gives rise to the outer covering of the animal, and we can see in this figure that this outer covering is the ectoderm. So in diploblastic versus triploblastic, we can see the difference of the mesoderm. Mesoderm, which is present in the triploblastic but is absent in the diploblastic. So endoderm is the innermost germ layer, and since it is the innermost, it gives rise to the lining of the digestive tract or cavity and organs such as liver and lungs of the vertebrates. This is the endoderm, which is being represented here in the black color. So this endoderm is basically the inner layer, inner germ layer, and it gives rise to the organs like it gives rise to the lining of the digestive tract or organs such as liver and lungs. If I talk about diploblastic cnidarians, in cnidarians and few other animal groups, we have two germ layers. As I mentioned, they are diploblastic. So in such cases, they have their cavity filled with a spongy fluid, which is known as the mesoglia. Although they do not have mesoderm, but they have a fluid-filled matrix, which is known as the mesoglia. This is important to remember. What is mesoglia? It is present in diploblastic animals. It is a fluid-filled matrix, which is present in the diploblast. While if I talk about the triploblastic animals, they have a middle layer, and it is present in all bilateral symmetrical animals, which have a third germ layer, which is called as the mesoderm. This mesoderm fills much of the space between the ectoderm and endoderm, and therefore it fills up almost all the spaces between the outer and the inner layer. So in bilateral symmetrical animals, the mesoderm forms the most of the organs like muscles and the most other organs between the digestive tract and the outer covering of the animal. So we know that the outer covering is being formed by the ectoderm. Endoderm forms the lining of the digestive tract, and therefore all the organs which are being present in between the outer covering of the animal and the digestive tract is being formed by the mesoderm, which is the middle layer. This was about the division of animals on the basis of germinal layers. So on the basis of germinal layers, I again repeat, animals can be divided into two groups, diploblastic and triploblastic. So diploblastic includes the organisms that have tissue level of organization, example cnidarians, while triploblastic includes the organ level of organization that have all the three tissues. You may also remember that what leads to the formation, like ectoderm is leading to the formation of what kind of organs? So ectoderm, since it is the outermost covering, since it is the outermost germ layer, it mostly gives rise to the outer covering of the animal. While endoderm, usually since it is the innermost germ layer, it gives rise to the digestive tract or cavity and organs such as liver and lungs of the vertebrates. And if I talk about the bilaterally symmetrical animals, which have third germ layer also, which is mesoderm. So mesoderm, since it is the middle layer, it fills up most of the space between the ectoderm and the endoderm. So we can say that all the organs which are being formed between the digestive tract, which is formed by the inner lining, and the outer covering of the animal is being formed by the of mesodermal origin. Next we have body cavity. So organisms can be divided into different groups based on their body cavity. So presence or absence of a cavity between the body wall and the gut wall is very important in classification. So the body cavity which is lined by mesoderm is called the coelom. So we learn about three different layers, ectoderm, mesoderm and endoderm. So if the body cavity is lined by mesoderm, it is called coelom. That is, it is called a true body cavity, right? The organisms can be classified into different groups based on the presence or absence of the body cavity. That is, they can either be coelomates, they can be pseudocoelomates, or they may have no cavity at all, example, which may be called as acoelomates. So coelomates and that possess the coeloms are called coelomates. Example, annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. In some animals, if I talk about the body cavity, as I mentioned, should be lined by the mesoderm. So in some animals, the body cavity is not lined by mesoderm. Instead, scattered pouches of mesoderm are present in between the ectoderm and endoderm. So such a body cavity is called as a pseudocoelomate. pseudocoelom, and the animals are known as pseudocoelomates. Example, SK helminthes. So we will see the figures of these coelomates, pseudocoelomates, and acoelomates later. So for now, coelomates are the ones which have body cavity which is made up of mesoderm, which is lined by mesoderm, while in pseudocoelomates, instead of the body cavity which is lined by the mesoderm, we have mesoderm as scattered pouches in between the ectoderm and endoderm. So basically, no true cavity is present, and therefore they are known as pseudocoelomates, that is false coelom. The animal in which the body cavity is absent are called acoelomates. So basically, on the basis of body cavity, we can divide the organism into three types: acoelomates, pseudocoelomates, and coelomates. One thing to remember is that in this classification, we did not include sponges at all, because we did not include sponges or cnidarians, because in those we do not have three germ layers. So this classification is basically meant for the organism that have three germ layers during their embryonic development. So this is how we have to relate each and every characteristic from one characteristic to another characteristic. Like body cavity cannot be present anyways in the cnidarians or in case of sponges, because there we do not have mesoderm. And body cavity is something which is known as coelom, which is lined by the mesoderm. Therefore, it should actually have mesoderm to be categorized into any of these three categories. So this is the diagrammatic section of coelomates, pseudocoelomates, and acoelomates. So in coelomates, we can see that. So in coelomates, we can see that we have ectoderm. We have ectoderm here. We have endoderm. We have endoderm here. And we have the body cavity. White space is the body cavity which is being lined by the mesoderm, right? So this is how the organism is called a coelomate. So the body cavity is being lined by the mesoderm, and the body cavity is known as the coelom. Now if I talk about pseudocoelom, so organisms that belong to pseudocoelomates, organisms that are pseudocoelomates are SK helminthes. So in SK helminthes, we can see that these scattered pouches of mesoderm are present. So this is the mesoderm pouches. So we have scattered pouches of mesoderm, right? So these are the scattered pouches which are present. Instead of a proper body cavity, we have pseudocoelom, that is false coelom. While if I talk about acoelomates, we do not have any body cavity between the endoderm and the mesoderm, and which is surrounded by mesoderm, and therefore they are known as acoelomates. Example, platyhelminthes. So in platyhelminthes, all the three germ layers are present, but the body cavity is not present. So they belong to group acoelomates. We can say that SK helminthes belong to the group pseudocoelomates. They have mesodermal pouches which are scattered into the body cavity. And while if I talk about the coelomates, they have the body cavity which is being lined by the mesoderm. So what are the functions of body cavity which is being present? So body cavity performs many functions. The most important function is that it's fluid which is being present in the coelom. It cushions the suspended organs and therefore helps them to prevent from the internal injury. So this coelom contains the fluid, coelomic fluid, and this fluid helps in cushioning the suspended organs, and therefore it helps prevent the internal injury. In soft-bodied bilaterians such as earthworm, the coelom contains non-compressible fluid that therefore, since the fluid is non-compressible, therefore it is hard and it can act as a skeleton against which the muscles can work. So this is the function of coelomic fluid in the soft-bodied bilaterians. The cavity also enables the internal organs to grow and move independently of the outer body wall. So the cavity actually allows the space, provides the space for the internal organs to grow and move independently of the outer body wall. So these are the functions of body cavity which are being present in the coelomates. So now the organisms, on the basis of how during the embryonic development if the cleavage is taking place, how the what is the fate of blastopore and how the coelom is being formed, organism can be classified into two different classes, that is the protostomes and deuterostomes. So during the development, cleavage is the first event, and basically cleavage is the first developmental event, and it is the cell division without the cell growth. So when the zygote is formed, it undergoes cleavage, that is, it undergoes cell division without the cell growth. So once the blastula is being formed, it undergoes cleavage. So in protostomes, the spiral cleavage occurs. So you can see in the figure that in protostomes, the cleavage is spiral. So this is the side view. While if we talk about the deuterostomes, the cleavage is radial, that is from the center, from the axis of the axis is the central axis. So it is the radial, and we can say that in the cleavage is spiral and determinate, and the cleavage is radial and determinate. So the fate of these cells is fixed, and the determinate in protostomes each can. So what do I mean by determinate? That each cell can contribute to development in only one particular way. Like there is, each cell can contribute to the development in only one particular way. While if I talk about the deuterostomes, the radial cleavage occurs and the daughter cells sit right on the, you can see that the daughter cells sit right on the top of the previous cells. Right? And the fate of these cells is indeterminate, that is, if they are separated from one another, it should be indeterminate actually. So if these cells are separated from each other, so these cells will independently develop to become the complete organism. While if in case of protostomes, the fate is determinate, that is, the fate is already determined, and each one can contribute to the development in only one particular way. While in case of deuterostomes, the cleavage is radial, and the cells, the fate of these cells, fate of the daughter cells which are being present on the top of the previous cells, they are indeterminate, and if they are separated from one another, each cell can go to become a complete organism. If I talk about the fate of blastopore, so animals with 3 tissue layers are either protostomes or deuterostomes. So this, whatever I am talking about, is for the triploblastic animals, right? So why they are named as protostomes or deuterostomes? So proto means first, right? Stomes means mouth. So if mouth is developed first, it is called protostomes. Deuter means second. Stoma again means mouth. So if it is if the mouth opening is formed at the second, then it is called as the deuterostomes. So during the development, a hollow sphere of cells, which is also known as blastula, is formed. So during development, one of the stages is the where the blastula formation occurs, which is the blastopore. So basically, as the development proceeds, a hollow sphere of cells is formed, which is known as the blastula, and the indentation, that is the depression that follows, produces an opening which is called as the blastopore. So a pore-like structure is formed in the blastula, which is known as the blastopore. This is the opening which is formed by the indentation or the depression of the blastula cells, and this is known as the primitive cut. And in protostome, blastopore becomes the mouth, and then the anus is being formed. While if I talk about the blastopore, in the blastopore, firstly the anus is formed and then it leads to the formation of the mouth. In protostomes, the mouth appears at or near the blastopore, hence the origin of their name. While in deuterostomes, the anus appears at or near the blastopore, as we can see that the anus is being formed near the indentation from where the cells are undergoing the depression, and only later does a second opening forms the mouth, and hence the origin of their name. So in protostomes, so basically protostomes and deuterostomes is a further classification of triploblastic animals where during the development, if the mouth is formed first, then it is called the protostome, and if the mouth is formed the second, that is, it is formed away from the blastopore, it is called as the deuterostome. Also, we talk about the coelom. So, coelom formation, we know that coelom forms by the splitting of the mesoderm, or coelom forms by an outpocketing of a primitive cut. So, how the coelom is being formed also results into the classification of animals into protostomes and deuterostomes. So, true coelom develops differently in the two groups. So, in protostomes, the mesoderm arises from the cells which are located near the embryonic blastopore, and a splitting occurs that produces the coelom. So, basically, it leads to the splitting, and then the coelom is being formed. So, coelom forms by the splitting of the mesoderm. While in deuterostomes, you can see that there is no splitting, but it is formed by the outpocketing. So, by the